Okay, so tonight we're here for the special senses lecture. So your special senses are going to be these senses with the more complex receptors, if you remember. And so because of that, that's going to include your vision, your hearing and equilibrium, your sense of smell or olfactory, and your sense of taste or gustatory. So these are kind of fun. This is kind of a fun lecture. It's interesting because the special senses are always kind of interesting and fun to learn about. So this is kind of a good one to end the semester on. And so again, they're housed in those complex sensory organs. That's why we give them their own special chapter, and that's what we call them, the special senses. You don't have to know these for the exam, but ophthalmology obviously is the science of the eye. And then otorhinolaryngology, that's a long one, is the science that deals with ears, nose, and pharynx or throat. So those are your ENT, as well as the larynx. Sorry, I left the larynx over there. So just a couple fun terms for you. So we're going to first start off by talking about your chemical senses, and that'll be the end of this lecture. And then I'll do a separate one for vision and a separate one for hearing and equilibrium because they're just a little more complex. But the chemical senses, olfaction and gustation, go together because they are going to have this interaction of molecules from the outside with these receptor cells. So olfaction, of course, is your sense of smell, and gustation is going to be your sense of taste, which in this case, if you look at this plate of cookies, right, you're technically going to do both. I mean, if you're eating the cookies, you can smell them and you can taste them. And you're going to see that these senses are complementary. They're going to kind of go together, and I'll talk about how important olfaction is into your sense of taste, gustation, and these two senses will kind of let us know whether a substance should be savored or it should be avoided. Both are going to project to your cerebral cortex as well, which is where your cerebral cortex will be where you will process that sensation is occurring. Okay. And then your limbic system is where you're going to associate your memories with either that taste or that smell. Like, ooh, that smell was associated with a good taste, or that smell was associated with a bad taste. And so that's where you're going to evoke those kind of strong emotional reactions to smell and taste. Now, chemoreceptors obviously are going to be used by these systems because they're detecting chemicals, either chemicals in the air for, sense of smell, or again, chemicals that are going to be mixed in with your saliva, okay? So these chemicals have to be dissolved in some type of aqueous solution. This kind of sounds gross, but they need to be dissolved in some type of aqueous solution to be picked up by our chemoreceptors. And what this means is for our nose, right, when we're breathing in these chemicals through the air, we need that aqueous solution that we call mucus, okay? So that mucus, one of the big purposes, there's lots of purposes, right, of that mucus, but one of the purposes of the mucus on our nasal membranes is to dissolve these, what we call odorants or chemicals that are in the air that are going to stimulate these nasal receptors for our sense of smell. So smell receptors are excited by chemicals that are going to be dissolved in our nasal fluid. So of course I just had to use a dog. It was a little less gross than a kid with the snotty nose. And then our taste receptors are going to respond to chemicals dissolved in saliva. And so of course this just was appropriate for those of you that have seen Turner and Hooch. I have a Great Dane, and she actually does this, which is pretty gross. But they have plenty of saliva, lots of saliva, to dissolve those, gustatory molecules. So for our sense of smell, we're going to do our sense of smell first, and then we'll talk about our sense of taste, which is gustation. So our sense of smell is, again, olfaction. And we are going to do this with our olfactory receptors. Now, our olfactory receptors are going to be located in what's called the olfactory epithelium. This is located on the roof of the nasal cavity it actually kind of is going to cover the top of the area of the superior nasal conchae. And it's going to contain, again, our olfactory sensory neurons, that olfactory epithelium. And so here, what the interesting part about our olfactory sensory neurons is they are actually bipolar neurons. So the sensory receptor is actually at the tip of this bipolar neuron. So our olfactory neurons, I'll show you here. Let me show you a picture of these. Here we go. Here are these, again, olfactory, our kind of primary olfactory sensory neurons here. And if you look, what makes them a sensory receptor at the same time is that they have these little olfactory cilia. And these olfactory cilia are going to be bathed in this mucus, right? That's lining the nasal cavity. And so that mucus will trap odorant, the chemicals in the air that will bind here and stimulate these olfactory cilia. So kind of interesting. So these receptors are actually first order neurons. We'll just talk about in just a minute. And then they do have supporting cells in the epithelial cells, the mucous membrane, that are going to surround and cushion the olfactory receptor cells because they're neurons. Again, they'll help feed those cells and things like that. We do have olfactory stem cells. This is kind of important because this allows us to produce some new olfactory receptor cells. So that's kind of important that we have the ability to produce some new olfactory receptor cells in case we damage some of those olfactory cells, which you can see that would happen, right, with our nasal, cavity being so exposed. So again, the receptors for these for olfaction are actually bipolar neurons, because here you can see again, the receptor is actually part of this neuron here. So pretty cool. Well, I mean, at least it just makes it pretty basic, right? And again, those bipolar neurons, oops, sorry, I lost my spot there. Those bipolar neurons do have those olfactory cilia that are going to stick down and actually bind to those, what we call odorants, okay? And those basal stem cells, again, produce the new olfactory receptors. So here you can see, just as a reminder, again, as air comes into the nasal cavity and it kind of weaves its way through the nasal conchae, at the very top of the nasal cavity here is where you're going to have these olfactory receptors. And again, these olfactory receptors are actually going to have axons that go through the cribriform plate. Remember that a long time ago, I know this is back in the beginning of the semester, but remember that cribriform plate in the ethmoid bone? So the reason that we learned about the cribriform plate is they have these little holes in them, right? And what goes through those holes are actually the olfactory nerve fibers. So that's how the olfactory nerve fibers get back to the central nervous system. So kind of interesting. So all those olfactory nerve fibers, as you'll see in just a minute, are going to meet in this olfactory bulb here. And then they'll synapse with the next neuron. So olfactory neurons, again, are these kind of unusual bipolar neurons. They have these olfactory cilia. They're kind of hanging off of the dendrites and sticking in the mucus and binding these odorant chemicals. And then the actual cranial nerve itself, okay, here, these cranial nerves here are actually bundles of these axons from those olfactory receptor cells. So that's basically what the cranial nerve, when we say cranial nerve 1, which is a bunch of cranial nerves here, these cranial nerve 1 are actually going to be these bundles of these non-myelinated axons that are coming off of those olfactory receptor cells. Oops, sorry, I'm jumping back and forth here. So that's all that is. Just, again, cranial nerve 1 is just the non-myelinated axons of those olfactory receptor cells. And olfactory neurons, again, are kind of unique for a neuron in that they have stem cells that give rise to new neurons every 30 to 60 days. Now remember, in the central nervous system, this doesn't happen. We don't have this ability to regenerate neurons in the central nervous system like we do here with olfactory neurons, and then you'll see also with gustatory neurons. Or gustatory receptors, sorry. So this is really unique. This makes, again, these olfactory neurons unusual that one, they're actually bipolar neurons. Their receptors are actually bipolar neurons. And two, that they have this ability to regenerate because they have their own stem cells. So basically we give rise to new neurons about every 30 to 60 days. So again, you can see what's going to happen is these olfactory cilia are going to bind an odorant molecule, okay? And then we're going to talk about when they bind that certain odorant molecule that they're designed for, it's going to obviously stimulate an excitatory potential. And if there's enough of an excitatory potential that's stimulated across all these olfactory cilia, then it can actually, again, end up stimulating an action potential, right, at the axon hillock here. And then that action potential is going to shoot back through the axon of this cranial nerve and then end up releasing excitatory neurotransmitter that will stimulate what's called the mitral cell. The mitral cell is going to be the second order neuron in this pathway here, okay? So we got our first order neuron and our second order neuron. And this synapsing that is occurring between the olfactory neuron here, the olfactory sensory neuron, and the mitral cell is occurring in the olfactory bulb. That's why this olfactory bulb is so big. It's got all these cell bodies here for the mitral cells. Now, when we actually get into kind of the specificity of olfactory receptors, when we look at smells, if you have any type of smell, it can contain hundreds of different odorant molecules that are kind of put together to make that certain smell. And humans have about 350 different types of odorant receptors. Now, this is something like a dog that has a much better sense of smell than we do. Obviously has even more of these odorant receptors. That's why they can detect things that we can't even smell. Now, each of these odorant receptors is going to respond to one or more odorants. Okay, so they can respond to, sometimes they can be stimulated by odorant molecule 1, or they can be stimulated by odorant molecule 2, or maybe even odorant molecule 3. Okay. And now each odorant can actually bind to more than one receptor type. So, odorant molecule 1 might be able to bind to different receptor types that are going to give a slightly different message for a slightly different smell, or kind of being put together for that sense of smell. And then each receptor cell has only one type of receptor protein. So what this means is each receptor cell is only going to send back a signal for a certain, type of smell. Maybe it's like very floral, or so each of those receptors is going to be very specific. So I know this gets kind of confusing. I'm not going to get, honestly, too much into the nitty-gritties of this. But really what I want you to know here is that this one right here, that each receptor cell is responsible for like one part of your sense of smell. So maybe that's like kind of the more floral part of it. Okay. And that just gives us that specificity with our sense of smell. Now, obviously, our sense of smell is going to be kind of these combinations, like each smell that we have is going to be combinations of different odor receptors. So maybe, smell number 1 that smells like cinnamon is going to be, activating odorant receptor 1, 5, and 8. And, smell number 2 is going to be, 5, 9, and 10 or something like that. And that gives you, more floral smell. I'm making this up, but just to give you an idea of how this works, and that's oversimplifying it, obviously. But that our sense of smell is a combination of those different receptors that are stimulated. And then we also have pain and temperature receptors that are in our nasal cavity. And these are kind of important because they're going to be important for responding to irritants. So again, we want things like these pain receptors to respond to something like ammonia, that maybe burns our nostrils, or I don't know how many of you have ever been cleaning with bleach, and it was just a little too intense and it burned your nostrils. That's stimulating those pain receptors that are telling you hey, this is an irritant, maybe you shouldn't breathe this in. It might not be good for the rest of your body. So that's kind of a protective mechanism for us. Also, we have these kind of temperature receptors in our nasal cavity that will give us this sense of smell of hot or cool, like that chili peppers kind of smell hot or spicy, and menthol would smell kind of cool. So just kind of interesting that we have some of these different receptors in our nose that help to give us these different smells. This is, again, just for fun. If I was going to ask a question on this, it would be purely extra credit because I know it's a dog's, not human anatomy, but you know that I love my comparative anatomy. So this one's a fun one. So obviously olfactory in dogs is highly pronounced. And that's because they possess up to 300 million olfactory receptors in their nose. This is compared to our only about 6 million in humans. So and that's a huge difference. They have way more olfactory receptors. And not only do they just have more olfactory receptors, but the part of a dog's brain that's devoted to analyzing smells is about 40 times larger than ours. So they have a very large portion of their brain devoted to smell. I mean, their sense of smell is a really big part of their awareness of the world. Now, dogs also have something called neophilia. Neo means new, philia means love of. This means that they're attracted to new and interesting odors. It's almost like an obsession for them. And that's why when you first walk into, someone's house and you greet their dog, they're obsessed with smelling you. And that's because of this neophilia. And that's because, again, that's a main part of how they sense their world. Now, what's really interesting is dogs can smell things that we can't, right? They have olfactory receptors that we don't have. And so this actually allows them to detect things in our bodies that we produce as a byproduct of diseases like cancer, and also even infections like COVID-19. So they're actually using dogs now and training dogs to be able to do things like sniff out cancer. Sometimes they can do this prior to someone even being at a point that they could be diagnosed for cancer. They, just wouldn't have hit a point where they would go in for that. Also, they found that dogs are able to detect changes again in, hormones and things like that. I actually just recently spoke to someone that's going to be going through school to do this type of training. And she said she's doing it mainly for individuals that have epilepsy. And what they do is they actually, when they have a seizure, they have to take off their underwear, it sounds strange, and put it in a bag. And they use that because you release certain biochemicals when that individual's having a seizure. And it really comes out in your mucous membranes. And so they use that smell in your underwear to be able to train the dog to that smell of, associate that smell of when you're having a seizure. And the dog then can oftentimes warn the owner because they can detect that slight change in body chemistry that happens right before you have a seizure. And they can actually alert the owner to the fact that they're going to potentially have a seizure. So, it's amazing that dogs can do this. And then this is just a fun news article because they do know that dogs can detect COVID-19 infections in humans. They actually use dogs at some event here, I think it was in California, to kind of survey the crowd to make sure that, individuals weren't coming in sick with COVID-19. So kind of interesting use of that there. Okay, so moving along here. Again, when I kind of already talked about this, but when these olfactory hairs bind an odorant in that mucus, again, it's just going to cause an excitatory potential. It's going to cause sodium channels to open. It's going to have, again, causing a depolarizing effect. And if there's enough depolarization that occurs, then it can trigger an action potential at that axon helix. And then that nerve impulse is going to be triggered, and it will, again, move back and synapse with the mitral cells here, and again, transmit that message on to the mitral cells in the olfactory bulb. Okay, so then from there, those impulses from the mitral cells are going to travel back via now olfactory tracts all the way to the olfactory cortex. And the olfactory cortex is really going to be where we're going to consciously be aware of and interpret that smell. Okay, so that's happening in our olfactory cortex. And I know we talked about the olfactory cortex in the brain chapter, but we didn't really do a whole lot with it. And that's because, again, it was part of the special senses. So this is where that comes into play. So some of that information, again, is going to be sent on to the frontal lobe. Some of that is going to pass through the thalamus, through that relay center, on up again. And as it goes through that relay center, it can split, and some of those, again, pathways will take us to places like the hypothalamus, the amygdala, and the limbic system. So why is it that, we're going to the olfactory cortex, where we're really kind of doing our conscious interpretation and awareness of that smell? That's the olfactory cortex. But then why are we going on to the hypothalamus, amygdala, and limbic system? And these are the places that we get our true emotional response to odor. Okay, so again, maybe you smell something that, like fire, that gives you this sense of fear. That again is being sent to the amygdala. Okay, or kind of, again, gives you that sense of fear, like, oh, I need to, do something stimulating your fight or flight system. Or it might go to the limbic system because you smelt like cookies that reminded you of your grandma, or a bakery that, you remembered as a kid, and it gives you this memory of a kid. So that's where your emotional, in the limbic system, that's where your emotional responses are going to come to that odor. Okay. So though that's an important part of olfactory. Now, this is kind of interesting because when I first started teaching anatomy, I think, gosh, it was maybe 10 to 12 years ago, I had this slide, but it was a very different slide on the vomeronasal organ. And we called it this other olfactory sense organ in humans that detected pheromones. And I gave you all kinds of examples of some of these crazy, funny studies where they had these college women come, and they smelled these sweaty shirts that they had guys these guys work out and put these shirts on, these white shirts on, and then have them work out and get all sweaty. And then they put those sweaty shirts in bags. And then they had, at separate time, they had women come along and smell these shirts. And so basically the kind of study showed that women thought that the shirts smelled better. They rated the shirts as smelling better when they were ovulating. And so they associated this with this vomeronasal organ. Well, what ended up happening is they kind of found out that this research wasn't all that great. It was actually supported, I think, initially by a perfume company that actually was producing this new perfume with pheromones in it. And so they found a lot of holes in the research. And so they've kind of come back around and done this. And the reason that we initially looked at these vomeronasal organ, the vomeronasal organ is just this organ that we have. It's right here, or also called the Jacobson organ. In many animals, we call it the Jacobson's organ. And it does detect pheromones in other mammals. Now, in humans, it ends up being in adults, it's just kind of consists of just these pits that we don't think really probably do anything. They're kind of like a vestigial structure, a remnant of like our kind of ancestral past. Now, what's interesting is the VNO, the vomer nasal organ, is present in the fetus, but it atrophies right before birth. And so we think that if humans are actually responding to things like pheromones, it's most likely that, sorry, this is supposed to say pheromones, not hormones, but it's most likely that they're using their normal olfactory system. So that's why you don't find this in your textbook anymore. You used to. And again, this is why we're constantly updating science. But so we think it's more of a vestigial structure now. So if you want to find more information about this, you can go to this article. It's kind of interesting. It talks about the VNO. So pretty much we still don't know. It's kind of there's not enough information for us to really kind of discern this. But you will hear about the vomer nasal organ or Jacobson's organ in other mammals. And it does detect pheromones in other mammals. So lastly, anosmia is kind of our clinical application of this. Many of you probably have heard of anosmia now because of COVID-19, and it just simply means without smell. So this can result from a number of different things. It could be head injuries that actually tear the olfactory nerves. It could be the after effects of nasal cavity inflammation, as you see in COVID-19. So many of the individuals that lose, I was actually one of these individuals that lost my sense of smell. I completely lost my sense of smell. I had, again, the complete anosmia. And it was just due to all of that inflammation surrounding those olfactory nerves. And then it can also be an indicator of certain neurological disorders that this is going to, again, be damage to those sensory neurons themselves. And that can be an indicator of things like Parkinson's disease or even things like multiple sclerosis. So again, but many of the individuals that, like me, that had the anosmia with COVID-19, I'm going to talk about this just because it's kind of the current thing that we're talking about with anosmia. Many of those individuals, like me, get their sense of smell back when the inflammation subsides. So I did regain my sense of smell, but I will tell you my experience in terms of its relationship with this next sense, which is our sense of taste or gustatory. So again, taste is another chemical sense. It's detected with, again, molecules that have to be dissolved. So this time, instead of dissolved in snot, right, dissolved in our mucus, they're dissolved in our saliva. And we have different taste receptors. There's going to be one for sour, sweet, bitter, salty, and what's called umami. And then we believe there's also a sixth one now for lipids. So let's talk a little bit about these 5 classes of stimuli. Again, sour, bitter, sweet, salty, and umami. Any other taste that you may have is usually a combination of these 5 taste sensations plus olfaction, okay? And so we're going to talk about olfaction is really important to your sense of taste. And if you had the anosmia with COVID-19, you know how important that is, because if you lost your sense of smell, your food probably tasted really bland. And if you want to try this out for yourself, I want you to take some cinnamon and sugar and then some cinnamon. And what I want you to do is put a blindfold on and then have, a friend hold one that's just, hold a cup that's just sugar and hold a cup that's cinnamon and sugar. And then I want you to just taste it with your finger, each one taste it with your finger and see if you can tell which one has the cinnamon in it. Oh, and plug your nose the first time. The first time you do it, plug your nose. The second time, go ahead and smell it first and then taste it. And it will shock you because I've tried it a couple times and it's amazing. Many people cannot taste that cinnamon unless they can smell it too. So it'll just give you an idea of how important your sense of smell is to your sense of taste. We do have about 10,000 taste buds that are found in our tongue in the form of these papilla. They're these little tiny kind of peg-like projections that you see here. Some of them are bigger, some of them are smaller. And then we're going to talk about the different types of papilla we have. You have fungiform papilla. That's kind of the majority of like the papilla that we have here. We also have what are these things here called the valate papilla or circumvallate papilla. And then we also have these foliate papilla that we'll talk about on the sides of the tongue here. So we have these different types of papilla. And then we do have 3 different types of gustatory cells. So we have our main receptor cells. This time they are not going to be a first order neuron. It's just the receptor cell. And then we do have supporting cells that again are going to kind of cushion those receptor cells and then also again provide nutrients and things like that. And then we do have these basal cells again. Just like in our sense of smell, our basal cells are going to act as these stem cells that will provide us the next generation of our taste receptors. So we do have the ability to replace our taste receptors if they're damaged. And you guys probably know this if you've ever like burnt your tongue, like if you drank something that was too hot, hot coffee, and it kind of, you damage some of those receptors, you know that like you just wait, you know, a week or, you know, so, and then all of a sudden your sense of taste is back. Those receptors have been replaced. So again, our taste buds are our sensory organs for taste, and we have these different papilla. Like we talked about, we have the fungiform papilla, which you're going to see here. They're scattered throughout the kind of surface of your tongue here. And there's kind of mushroom-shaped structures. These house the majority of our taste buds, and that's why, again, they're scattered all through the top of our tongue there. Then you have the foliate papilla, which are on the sides of your tongue here. And then you have the valate or circumvallate papilla. I apologize that I must not have, I accidentally left out the grouping for this. So if you want, you can put a tag here and just put valate papilla. These are these ones that form this V back here. They're large papilla. They're the largest of the papilla, and they form that V in the back. And then we do have a few papilla on like the soft palate and inside of our cheeks, even the back of the throat, like in the pharynx and the epiglottis. And you probably know this if you've ever like accidentally, you know, gotten like Listerine or something like that, kind of too far back here, and you're like, whoa, it burns. That's because again, you're stimulating some of those pain receptors, which are again one of your taste buds back here in the back of the throat. So you do have some taste receptors in the back of the throat, and again, some of those are protective to make sure you're not swallowing things that you shouldn't. There are again our 5 basic taste sensations. And so you're going to see that these taste receptors for each of these sensations are going to be scattered throughout the tongue in the same places. So you will have for each taste receptor, you're going to have them scattered here in the fungiform papilla, here in the folate papilla, and here in the valate papilla. Okay. So you'll see you can taste those sensitive tastes throughout your tongue. So we have sweet is the first one. It's going to respond to things like sugars, saccharin, even alcohol. Some certain amino acids will stimulate that sweet sense. And then lead salts, believe it or not, will also stimulate that sweet sense. Sour is actually going to be stimulated by hydrogen ions in solution. So technically your sense of sour is really going to be associated with things that are acidic. So kind of interesting really what our sense of sour is. And then salty is going to be responding to metal ions. So like inorganic salts like sodium chloride, right? And so that's why sodium chloride gives us kind of the strongest sensation. So that's why it tastes the saltiest for us. Bitter is going to be primarily alkaloids such as quinine and nicotine, caffeine. That's why, you know, if you drink coffee, that caffeine can taste a little bit bitter, and non-alkaloids such as aspirin. Now, a lot of your bitter taste is associated with things that are alkaloids that are going to tend to be more basic. Not always, but will tend to be more basic. Now, you may say, well, caffeine's not basic because my coffee is acidic. But remember that the caffeine itself isn't acidic. It's the other things that you have in your coffee that make it acidic, as well as your response, your sympathetic response to caffeine. And that sympathetic response shuts down some of your digestive, parasympathetic system. And so what that does is that kind of ends up allowing some of your stomach acid to kind of back up as you slow down your digestive tract. So sometimes, what we associate with an acidic food might not necessarily be, the specific chemicals that are in there. It might be other things. So kind of interesting there. So those are your bitter things. And then umami. Umami is kind of what we consider our savory taste. So this is going to be amino acids like glutamate and aspartate. You'll find these in meats, a lot of meats and cheeses. And we originally kind of came up with this sense when we were studying monosodium glutamate. So this is called the fifth gustatory sense because it was one of the last ones that we discovered before I'll talk about this new sixth sense that we think we have. And so, originally we said it was the sense that I taste dead things. So it was detecting glutamate that we find again in a lot of meats. And it's believed to arise again from these taste receptors that were stimulated originally by monosodium glutamate, MSG, that we often add as this flavor enhancer. And that gives us this savory or meaty flavor, okay? So that's your sense of umami. Then we now think that there's a possible sixth taste, and this might be why we crave, lipid foods with lipids. So there's growing evidence that we can taste these long-chain fatty acids from lipids, and that perhaps this is our sixth taste receptor, and that may explain why we like fatty foods. So that's kind of an interesting one. You have to kind of watch as that develops. But there's some pretty good research now kind of supporting this sixth taste receptor. Now, keep in mind that a lot of our taste likes and dislikes do have some type of homeostatic value, meaning they're trying to help us maintain homeostasis. They basically help us guide the intake of certain foods and things that would be beneficial, and then potential substances that can be harmful. So, for example, our dislike for things that are really sour and really bitter could be because oftentimes some of those really sour and really bitter things can be poisons. Really acidic, really basic, again, are going to be harmful to our body. So this is a protective way of warning us if something, is poisonous, or even if something is spoiled. It will often, again, stimulate something like sourness or bitterness. Now, really quick, we'll talk about the anatomy of the taste bud, which is pretty basic. So when you look at your taste bud, you have this oval body in your taste bud that's going to be filled with these gustatory receptor cells. So here are going to be these gustatory receptor cells, and they're going to, again, be in contact with this first order sensory neuron. So this oval body consists of about 50 receptor cells that are also going to be surrounded by these pink cells here that are called the supporting cells. And those supporting cells are going to kind of cushion those taste receptors, and they'll also provide, some nutrients and things like that. They do the support. They're the support cells for those gustatory receptors. And then the gustatory receptors, if you look here, they're going to have these little gustatory hairs. They're technically microvilli, but we call them gustatory hairs that project off of these gustatory receptor cells into what's called the taste pore. That would be bathed basically in saliva. And so again, as that saliva is moving over, it'll bring in those different, gustatory chemicals, or just different chemicals that are in your food, and they can bind to and then stimulate an excitatory potential in those gustatory hairs. Okay? Also, then we have these basal cells. So you'll see down here at the very bottom, we have these little basal cells. Those are kind of our stem cells, and they'll develop into new receptor cells every 7 to 10 days. So that's why typically if you end up kind of burning, your mouth in terms of you drank a really hot coffee and you're like, oh, I damaged some of my taste receptors, I can feel it, then you usually get that back within about a week, because again, 7 to 10 days, you're going to be producing new receptor cells. Kind of interesting. So again, in these receptor cells, remember that because they're receptor cells, we're just producing receptor potentials, no action potentials. Now, if they bind enough of the right chemical here, and it causes enough of an excitatory potential, then they will release neurotransmitter that's excitatory and then cause an action potential in your first order sensory neurons. Okay? So that's all basically this is saying, is those graded potentials that develop in the gustatory hairs that can cause the release of neurotransmitter that give rise to those nerve impulses in the first order sensory neurons. And those first order sensory neurons, again, are synapsing with those gustatory receptor cells. So that's all of what we just talked about. Now, what's interesting is you have pretty rapid adaptation of taste. You also have pretty rapid adaptation of smell as well. So your sense of smell, you may like walk into a room and you find that something is really pungent. You're like, whoa, that's a bad smell. But within just a few minutes, you don't notice that smell anymore. And so that is because we have pretty rapid adaptation of these receptors, both your olfactory as well as your taste receptors. So with taste, you can get complete adaptation in about 1 to 5 minutes. And so those receptors become less sensitive to those chemicals if they're continually exposed to them over 1 to 5 minutes. So for example, if you're doing a wine tasting, it was interesting, and I don't have the graph of the data anymore, but what they found is when people took that first, drink or 2 drinks of the wine, that they could detect all of these, all of these different kind of tastes within that wine. But as they drank the wine, it kind of became more bland by, the couple minutes in. And so that's because of this rapid adaptation in our receptors. Now, obviously, different gustatory cells are going to have different thresholds for activation, and things like your bitter receptors, one, are going to be more sensitive, which just means that you require less of that chemical to actually stimulate that excitatory potential and eventually action potential in the first order neuron. And that's a protective thing, because obviously we want to very quickly stop eating that because it could potentially be a poison. And then there are other receptors that are going to adapt more rapidly. Okay, so things for some of your like more rapid and less sensitive receptors are going to be things like for sweet and salty. Okay, because those obviously are not likely to be very harmful. And so all of our gustatory receptors are going to adapt in about, they'll start to adapt, meaning they'll start to become less sensitive in about 3 to 5 seconds, with complete adaptation, again, anywhere between 1 to 5 minutes. So pretty quick, pretty amazing. And again, that's why food becomes kind of more bland as you're eating it continually. So our gustatory fibers are going to enter a couple cranial nerves, and this is a good chance for me to review some of these cranial nerves. So remember that one of the first cranial nerves that first order neuron is going to make up, so the axon of that first order neuron is going to make up cranial nerve number 7 if it serves the anterior 2/3 of the tongue. So think facial. Again, remember, facial is going to be sensory neurons from the face. So again, you can think of that sensory for the first 2/3 of the tongue, okay? Actually, I'm sorry, and I said that wrong. So remember the facial cranial nerve 7 is mostly motor to your facial muscles, but it has this kind of side little sensory portion, and that's just for the anterior 2/3 of the tongue. So remember, think of the facial cranial nerve as serving mostly motor innervation for controlling the muscles of the face, but then it has this little side sensory for the anterior 2/3 of the tongue. Okay, so think facial, 2/3 of the tongue. Then cranial nerve number 9 is the glossopharyngeal. So remember, glosso is tongue, pharyngeal. Think pharyngeals towards the back of the throat, right? Your pharynx. So that might help you to remember it's the posterior 1/3 of the tongue. So towards the back of, again, the tongue, because it's got that pharyngeal in there. So think towards the back, posterior 1/3 of the tongue. And then the vagus, I wouldn't touch on this. Vagus, cranial nerve number 10, sort of the palate and epiglottis. It's kind of more of a smaller function of the vagus nerve. It does many other important things. So again, I wouldn't test you on that so much. But I could test you on these two because they're a big portion of the gustatory pathway. Okay. So again, information will come in those first order neurons through cranial nerve number 7 and cranial nerve number 9. And then it can, again, be sent through the thalamus, that relay center, onto places like the limbic system and the hypothalamus. Okay, remember, it's going to go to the limbic system so we can associate that emotional response with that taste. And then it will move on to that primary gustatory area, just like, again, olfactory moved onto that primary olfactory area in the cerebral cortex. Now we're going to the gustatory area in the cerebral cortex, where we're going to get our conscious perception of that taste, okay? And again, those side shoots of the limbic system are going to give us that emotional attachment to that taste. So again, this is just kind of showing you that, going through, again, going up through the thalamus and then into that gustatory cortex. And then, again, you'll have offshoots that go into the limbic system. So now keep in mind that taste is actually 80% smell, meaning that this information that we're taking in with our taste buds is combined in our brain with our sense of smell to give us our overall sensation of taste. And without smell, again, our foods are going to taste really bland. If you experience the anosmia with COVID-19, then you experience this, because I did. My food was bland. This was a big one for me. My morning coffee just didn't taste as good. It was bitter. It just wasn't as flavorful. And so when I got my sense of smell back, I really enjoyed tasting my coffee. But yeah, many of my foods were really bland. And again, that's because taste is actually 80% smell. Try doing this. Try starting to taste some of your foods and, pinch off your nose, taste that food, and then smell it and taste it, and you'll have a totally different sensation. Interesting. Also, thermoreceptors, mechanoreceptors, nociceptors, these other senses that are not part of our special senses that are in our mouth are basically our touch receptors and things like that in our mouth, are also going to go and influence our taste as well. So temperature and texture can enhance or even detract from our overall sense of taste. So kind of interesting. Okay, so I'm going to go ahead and stop here, and then I'll start the next lecture on vision.